A lot of our research now is going into trying to come up with machine learning tools that can analyze MRI scans of any resolution and contrast.
Machine learning and artificial intelligence apps have been used in medicine and life sciences for a while now. And the project NextBrain that you heard my guest Eugenio Iglesias talking about just now is one of them. And together with one of the developers on the project, James Hughes from the University College London, we explore what makes this project special. One of the pleasures of being a research software engineer is that you get involved in science projects and in areas you haven't worked in before. And in the course of doing that, learn a lot of new exciting stuff. And so it was with my ex-colleague James Hughes from the University College London and myself when we embarked on a project called NextBrain. Our job, as James will explain a little bit later, was to come up with a web browser-based app to allow users to navigate through a brain and be able to zoom into individual regions showing anatomical details in high resolution. But NextBrain, the project, involved a lot more than that. The project is, if you forgive the pun, the brainchild of Eugenio Iglesias from Boston in the US. A project that started some 10 years ago, a time when the AI and machine learning landscape looked very different to what it is today. And this is a subject we touched on in our conversation. But apart from using deep learning tools, what fascinated me about this project was the chance to look, if only briefly, into the fascinating world of neuroscience and exploring the human brain. Both James and I learned a lot from this. Well, the project has now come to an end, and the work has just been published in Nature with the title A Probabilistic Histological Atlas of the Human Brain for MRI Segmentation. A perfect opportunity, I thought, to reconnect with Eugenio and James and to dive into NextBrain once more. And let's start with Eugenio.
My name is Juan Eugenio Iglesias. I was born in Seville too long ago. I went to college in Seville, in my hometown, in southern Spain. My major was in telecom engineering. It's a long story, but I wound up in Sweden with a grant from the Swedish government doing electrical engineering. Went back to Seville as a research assistant, applied for PhDs, lived in Denmark for a while, and then moved to California. I did my PhD in biomedical engineering at the UCLA. Then moved to Boston for a postdoc at Mass General Hospital. Met Caroline, who's now my wife, got kicked out of the country. I didn't do anything bad. It's just my visa expired.
Okay.
I had a 2-year requirement to live in Spain, so we moved to wonderful and beautiful San Sebastian in the Basque Country. It's the city, this is a completely unnecessary plug, but it's the city in the world with most Michelin stars per inhabitant. Not that I go to Michelin star restaurants, but the food is amazing. So I highly recommend it.
I didn't know that. Well, I heard about San Sebastian, but I didn't know about the Michelin restaurants.
Oh man, it's amazing. Yeah. And then from there we moved to London, where I worked at UCL for a few years, as you know. And then we ended up moving back to Boston a few years ago. So I'm now associate professor here at Mass General and Harvard Medical School. And yeah, this is me.
What a story. I didn't even know half of it because we used to work together on this project called NextBrain at UCL at a time when you were already, I think, in Boston.
Correct. Yeah. Yes, we tried to settle down in the UK. It didn't work out. Brexit didn't happen. So I'm not complaining about Brexit. Maybe I am, maybe a little. But things didn't work out professionally. So Caroline ended up moving back here. So I followed her.
NextBrain actually is a project, well, as the name suggests, in neuroscience. But you have a degree in engineering. So did you then switch in that career that you just described to neuroscience? Or are you still sort of kind of retaining your engineering hat on?
Oh, for sure. Yeah. I'm an engineer. My PhD was in biomedical engineering, but I did have to take some classes in anatomy, neuroanatomy, neuroscience, and so forth. But most of the classes, most of the electives were like optimization, neural networks, and stuff like that. It's just that I love making software to study the brain. I'm fascinated about the human brain. I'm not as good as a neuroscientist in neuroscience, and I don't want to be. I don't need to be. I just know enough that I can talk to them, understand their needs, and create this software, these tools that they can use to study the brain and do cool stuff, like NextBrain.
Like NextBrain, indeed. So what is NextBrain, sort of in a nutshell?
Sure. So NextBrain is a so-called probabilistic atlas. So you can think of it as a 3D image. Like a normal image has for every pixel, right? You have an intensity, you have a brightness, right?
Yeah.
A probabilistic atlas does not have a brightness. It has a bunch of probabilities for different labels of brain regions. So, for example, if you are in the center of the white matter, maybe that the probabilities say, oh, this is 100% white matter, and 0% everything else. But maybe if you are right in between the white matter and the cortex, it says 50-50 or something like that. Right? For every pixel, instead of having a brightness, you have mathematically speaking, you have a long vector with probabilities for all the different brain regions. Normally, most of them are 0. It's only like 2 or 3 or maybe 4 that are non-zero, depending where you are. And then the way you build this atlas is essentially by taking a bunch of brains from different people. You need to label them because you need to know where things are. And then you essentially align them, you co-align them together, put them on top of each other and average these labels. So people did this for a very long time with MRI, with 3D MRIs of living people. What makes NextBrain special is that we did this with very, very fine-grained labels that we derived from histology, from microscopy. So instead of using brains from living people, images obtained with an MRI, what we do is we take brains from donors, we slice them, look at them on the microscope, get much more accurate and fine-grained labels, and then build the atlas from that. And that's what makes it special.
Because the slices themselves are thinner than a slice you get from an MRI scan. Is that what it is?
Yeah, it's not only that the slices are thinner, is that you can actually put them in the microscope and scan them, right? You see so much more than you would see from the MRI. The resolution is so much higher. And also because you can use these stains, you have to use stains to highlight different types of cells. You can essentially highlight whatever you want, so to speak. So you can see a lot of things that you cannot see with MRI.
Oh, okay. So what kind of different cells are we talking about? So we probably know about neurons. Maybe some people know about axons. Then there are the glia cells.
Oh, man, yeah. This is a mess, right? So NextBrain was published in Nature. In the same issue, they published a bunch of papers from the BICAN, which is the Brain Initiative Cell Atlas Network. They're trying to build a census of types of cells we have in the brain. So it's really messy. We don't really know the whole story yet. This is an area of active research. So we are trying to figure out what type of cells we have and where. It's still TBD.
Okay. Now, I thought that we kind of settled that, but apparently not, which is good to know. So there's still things to discover. So we do know about white matter, though. So what is white matter?
White matter is essentially all this stuff in the center of the brain where we have all these fiber tracks connecting the different regions. So the white matter is has all these highways connecting different regions in the brain. People have known this for a long time from dissections. Whereas the gray matter comprises the cerebral cortex, so the shell, so to speak, and a bunch of nuclei that are also deep in the brain. So that's the main distinction. To study the white matter, people use this tool called diffusion MRI, where for every pixel, instead of getting an intensity, as you would get with a normal MRI, like a brightness, what you actually get is a distribution of the orientation in which water diffuses, which is a putative measure for fibers. So if you have, for example, in a given voxel, you have a profile that says, oh, water can diffuse in every possible direction. Yeah. That means it's probably water. Well, in the case of the brain, that's probably cerebrospinal fluid. Water can go anywhere unrestricted. Whereas if you have a profile where it's oh, no, water can only go in one direction, so much more than in the others, it means you're probably in the middle of a freeway going in that direction, right? So say the corticospinal tract or the corpus callosum, right, which is the big freeway between the left and the right hemispheres. If you look at it with diffusion MRI, you have this super anisotropic profile. But again, for every pixel, you don't have one number. What you have is a distribution depending on the direction you look at.
This is truly fascinating.
It's super cool. It's crazy we can do this. And this is,
absolutely, yeah.
people use diffusion in the beginning mostly to do so-called tractography, where you say like, okay, if I have a pixel or a voxel in 3D, like I said, you have like a spherical distribution, water can go either direction, then that's probably not a fiber tract, right? But if you have one voxel where you have a distribution that looks like a stick, meaning like, there's clearly fibers going this direction. Exactly. And then if you follow that stick and you have another stick more or less in the same direction, and then another one, then you can keep following, right? Because that's probably a fiber tract. And you can reconstruct these spectacular images of the brain. I don't know if you've seen them. People have like this super pretty, if not, you should totally Google it. Like full brain tractography. It's spectacular.
Now, I think I've seen that there is the optical pathway from the eyes, which goes first, I believe, to the back of the head, and then
occipital,
exactly. And the audio one, I think I might have seen as well. But I think you're right; it's absolutely fascinating.
It's absolutely staggering. And then, like a few years ago, actually pioneered at UCL by Daniel Alexander and colleagues, people started to use diffusion to look at the microstructure of the. I mean, it's really fascinating. I don't want to derail the conversation too much.
Yeah, we can stay there for a bit.
And if you can convince, he's very busy now because he's head of computer science now. But if you can convince Daniel Alexander to talk to you, it'd be amazing.
We'll find out about that tonight. I can reach out to him. So about NextBrain, so we're taking slices from donors, from dead people. So I think, as far as I remember, when I worked on it, I think there were about 3 or 4, maybe 5. I can't remember.
5, yeah. It was meant to be 10, but it was too much.
Well, indeed. But we also had MRI images.
Correct.
And I think, as far as I remember, we tried to locate what we saw on the slides with the position, as we can see, the kind of 3D visualization that you get with the MRI, and locate where that particular histology slice was in the 3D orientation of the brain. Is that correct? Is that my memory serving right?
Yeah, that is absolutely correct. Yes. The thing is, like I said earlier, we are trying to build this probabilistic atlas, right? To do that, you need labels in 3D. Of course, if you get an MRI, which is 3D already, and label it with a dedicated viewer, your labels will be in 3D already. But if you take the brain, you cut it into slabs, then you cut the slabs into blocks, then you put the blocks in the microtome, and then you slice them into sections. This is a lot of steps, a lot of that are like breaking the brain into a lot of like very, very thin sections, I should say. These sections are 25 microns thick, meaning that when you pick them up with tweezers and put them on the glass slide, they break, they distort, right? The shape is deformed. So you end up with this jigsaw puzzle, so to speak, where you have to take thousands of sections and arrange them in 3D together again to recover the original shape. Now, the reason why we took the MRI is like, we don't care, quote unquote, about the MRI per se, but the MRI is like the picture on the box of the jigsaw puzzle. So what we're doing is saying like, let's arrange all these sections such that they make sense with each other, but also with the picture on the box. In fact, the only reason why we can do this with decent accuracy is the fact that we know that we have all the pieces. We know that everything has to fit together. We know that the different sections in 3D, they cannot overlap. You cannot leave gaps in between either. And the global shape has to match the MRI. Now, the thing was, we did this to build the atlas, but having the core-registered MRI and histology of like full brains, it was so unique that we were like, we have to share this. People should be able to inspect this visually and play with it, because I think from the educational perspective was so interesting. And this is when you guys did a fantastic job helping us with the software and creating the online viewer. I'm assuming you're going to put the link on the description of the video.
Absolutely. Yeah, it's still there. I'm definitely going to put it up there. But let's return to the microscopy. You say the slices are 25 microns thick and they distort and they break. So how did you manage that? I mean, is that something that a human is doing, or is that actually something that a machine is doing?
No way, no chance. I mean, as humans, I think maybe if it was in 2D, you could do it to some extent, but in 3D, I mean, it's just impossible, right?
But I mean, putting the slices on a microscope and reading.
That's all human and that's all bananas. Yeah. Eventually, we only did a subset of select sections on the microscope because the data were too large. And for what we wanted to do, it was enough to have a bit less resolution. So the middle ground was to scan them in a high-end flatbed scanner. So the one you would buy for documents. It turns out that a high quality scanner will give you like a pretty good resolution, like about 4 microns per pixel, which is pretty darn good and definitely enough to see the cells macroscopically. And for what we wanted, which is like drawing these boundaries between brain regions, it was enough. And also for the viewer, it's much easier, right? For the online viewer. We do have some sections that we use for presentations and videos and stuff, where we start from the MRI, we switch to the histology, and we keep zooming in to the infinity and beyond, and it looks very, very spectacular, because at the end, like a pixel from the MRI is like a zillion pixels of the histology.
Yeah, I do remember that. And that was very impressive about the NextBrain thing. So the whole project has been running for a while. I think you did a big hoorah on LinkedIn when it got finally published by Nature, but it started 10 years ago, didn't it?
Correct.
So when and why did it start? So what got you into that?
Yeah, so when I came to Boston the first time as a postdoc, my postdoc supervisor hired me to build an atlas of the human hippocampus, not the whole brain, just the hippocampus, which is the center for memory and spatial navigation. And a lot of people are interested in it because a lot of neurodegenerative diseases are involved, for example, or most notably Alzheimer's and dementia. So we were doing this with super cool MRI of ex vivo, so of donors as well. So if you have a dead person, right, and they donate their brain, you can take the brain and chuck it in the scanner literally for a week. And then you can get ultra high resolution MRI, which is very nice. For me, it was great because I didn't have to deal with this 3D reconstruction problem. The images are 3D already. And you can do pretty good labeling on those. So that we built the atlas and it was pretty successful. People liked it. It's actually my most cited paper today.
Right.
And then we were like, this is cool. So my boss was like, okay, you should do the amygdala as well with collaboration with these other people. So we did it. And then what happened was when we switched to the thalamus, and this is when I went back to Spain to serve my 2 years, we realized that even on this ex vivo high resolution MRI images, there were boundaries that you didn't see that well and that were ambiguous. So we were like, why don't we use histology? This is not a crazy idea. There's people at UPenn that were trying to do the same thing. I mean, it's a bit crazy. There's not that many groups doing it, but we were not the only ones.
There are a few crazy ones.
Few crazy ones. I had this Marie Curie fellowship to build an atlas of the thalamus using histology. And we thought it was going to be easier than it was, but we got it to work eventually. And we learned a lot in the process, but we had to do a lot of things manually. And then I was like, okay, I really enjoyed this project. And I think it'd be really cool to extend it to the whole brain. But there are several things that we need to scale that cannot be like manual anymore. They have to be computational.
Right.
So that's going to require developing methods. Also, if we're going to do the whole brain, it's going to take years to acquire the data. So I applied for a so-called starting grant of the European Research Council, which is a 5-year fellowship with funding to build up your own team. It's meant to help postdocs or junior faculty transition into the next level.
Yeah.
So there is good money. It's 1.5, or it used to be 1.5 million euros. So it's good funding. Yeah, this is what I did at UCL. And we used that money to hire research assistants working at the brain bank, hiring postdocs and students working on the computational methods. It was a long process because acquiring the data alone was already 6 years. But
I
even when we,
so,
and it took a while to start. The funding was 5 years, but because of the pandemic, we got to extend it. And also, obviously, the pandemic also slowed down the data acquisition. I think we had the data at the end of year 7, but even then we were not fully ready with the methods. So it took a bit longer to get the atlas that we really, an atlas that we were proud of. And then, you submit to the journal, methods keep improving, you fix this, you fix that. And then at one point you're like, okay, this is it.
I do remember that when James and I started on the project, there was one patient data available. And I think the other ones were still in the making, or maybe, so it kind of evolved while we were working on it. So it was quite a long way in the making. But a project that long, the technology has changed as well, because I think over the past 10 years, we've seen really an explosion in deep learning methods or in methods in artificial intelligence and machine learning in general. I mean, how did that affect you and your team?
A lot. There's 2 separate aspects to this project. One was the data. Acquiring this data was a bit crazy, honestly. Like I said, we're not the only ones, but there's not that many labs doing this. And the other two labs that have been doing it, one is the Allen Institute, but they only did one. And they were like, okay, this is crazy. It's a beautiful brain, but they only did one. And then the only lab that does this regularly is Jülich in Germany. But they do have a dedicated setup for doing this. So we were the first ones who did it like with a normal microtome, without dedicated equipment and so forth. The data, we knew that was pretty unique, and we were safe, so to speak, on that end of the project. But on the methods side, I think at first we were all a bit freaking out. In terms, like the older, not everyone, but the older among us, right? We were like, oh my God, like our papers are going to be obsolete. And nobody's going to cite our papers. We're going to be so behind the younger people catching up with these methods because they grew up with them. But actually, it was the best thing that happened to us because we don't have to necessarily be innovators in this space. We can be consumers, and we are consumers. If you go to medical imaging conferences, the joke, well, half joking, half serious, is that it's essentially the papers from the previous year's computer vision conference recycled to medical imaging applications. So the fact of the matter is that the results got better thanks to machine learning, like the Atlas building, which required this. Well, first of all, reconstructing this jigsaw puzzle was definitely more accurate thanks to deep learning methods. So deep learning registration helped a lot. Second of all, the manual labeling was not fully manual. Because what we did was to say, okay, we're going to label one section every 4 and then use machine learning to fill in the gaps. And that was incredibly effective. We thought, we'd have to go back and, manually edit a little bit. We barely had to edit it. It saved so much time. Without deep learning, we could have used classical methods to do this, but the manual editing involved would have been like so much larger
and so much longer. Obviously.
Oh my God, would have taken forever. And the other thing is that once you have the Atlas, when you apply to automatic analysis, automatic so-called segmentation, so automatic labeling of new scans, it also works so much better, because now what we do is that if you give me a new brain that you want to label, I don't map the Atlas directly, but rather what we do is to use machine learning method to automatically segment the scan, not with resolution and the level of detail of the Atlas, like Corsair, but like in an incredibly robust manner that we couldn't dream of before. So, and you get that in seconds. So in a few seconds, you have like a super robust segmentation. It has fewer labels and less resolution, but it guides the mapping of the Atlas of NextBrain onto the new image that you want to analyze. So all these things work so much better and so much faster than they would otherwise. So it was great, super happy. I guess we were lucky in a way. This whole thing happened, but I guess there's always a bit of luck involved, right?
Yeah.
What is that quote about, success being the intersection of opportunity and, I don't remember off the top of my head. Essentially, you have to put the hard work, but there is always the luck factor, always.
I mean, deep learning is also an umbrella term for lots of different techniques and architectures. Did you have to change that a lot? Did you experiment a lot with that? Or how did it come about, and what kind of architecture are you using?
Yeah, in short, what happened is that by the time we started adopting deep learning techniques into the project, it was more or less clear, like everybody was using this UNet architecture that was invented. It was proposed in 2015 by this guy who was at DeepMind. Well, he wasn't at DeepMind at the time. He moved to DeepMind after, Olaf Ronneberger, which is super popular in image analysis. Essentially, the year he proposed UNet, he realized immediately how good it was. So he competed in every challenge that we have at conferences, and he was going around winning them all. But I think that was very nice, because people realized, like, okay, this is not an architecture that works for one problem because of the nature of this specific problem. This is like a very general architecture that does a great job, and everybody was using it. So we used it. It was doing great. And maybe 2018, 19, people started trying new things. And then transformers, of course, came about that completely transformed, no pun intended. They started with natural language processing. Essentially, attention, which is this mechanism at the core of transformers, is arguably the single most important trick in neural networks in language, and maybe altogether these days. In language, it really, really boosted performance by a lot. And then people got very excited, ported this to images. For natural images, transformers are really good. For medical images, it's like, I don't know. We were a
Not
bit,
very good.
we tried a little bit. No,
Yeah.
it's more like they're more expensive, but they don't really improve things that much all the time.
Mhm.
And we tried for a while. It didn't improve results that much. And we were like, we can stick to like the unit already was pretty good.
Mm.
Technically good enough for what we were doing. And even though we were open and we tried here and there, we didn't really switch. Funnily enough, there was a paper a few months ago, maybe last year. There is these guys in Germany that came up with the so-called NN unit, where the double N stands for no new. So they acknowledge that there is nothing new in here. But they did a fantastic job. They
Yeah.
essentially, these guys, what they did was these are like big unit fans. And one of the not a problem, but one of the things with units is that with time, they have all these bells and whistles. If you want to train a unit, you have to make all these decisions about how many levels shall it have. Shall it work in 2D or in 3D? Shall it have like residual connections or not? Shall it have this many features, or the compromise between depth and features? Like, how do I solve this? So these guys came up with a set of, it's a combination of heuristic rules
Yeah.
and a bit of trial and error. And essentially the way it works is that you give an end unit your data, and then it figures out the best architecture according to these rules and trains a segmentation network. Essentially, a bit like Olaf Runeberger did in 2018, these guys also won every possible challenge without, and it was beautiful because they said, we don't have to fine tune anything. We can just give the data to the algorithm, and the algorithm figures out what it should do. So these guys have been comparing an end unit to the fanciest, latest, and greatest transformer-based architectures in medical imaging. And essentially, they have shown that if you are fair in terms of allocating the same resources to both architectures, the results are the same, pretty much. Like the unit is good enough. Yeah, we played with others, but essentially we stuck to the right
That's what you start
architecture.
with. And what kind of tool did you use for the machine learning aspect? Was it PyTorch, TensorFlow, or what?
Yeah. So in the beginning, it was a bit of a mess because every postdoc had their own favorite software, right? I had a student that did everything in TensorFlow. I had two postdocs that did everything in Torch, but eventually Torch won the war. So since 2021 or so, it's everything is Torch. All the development we did, which again, it has not so much to do with the architecture, but more with like how you frame the problem and the type of techniques that you use to train, the types of losses, supervised, unsupervised, contrastive, yada, yada, yada. Everything was done in Torch.
So one of the questions, now that the paper is out, if we return to the research project itself, now that the paper is out, how do you think, or how do you envisage that NextBrain is going to be used? You mentioned educational use cases.
Yeah, correct.
Are there any other use cases you can think of?
Oh, yeah, for sure. Yeah. Okay, so there is several aspects. The first one is, of course, we have the data.
Right.
All the data have been made publicly available. All the links are in the website and the paper. So if you're a researcher in this space and you don't have the luxury of or you're not crazy enough to try to get funding and acquire this data for 5 years, you don't have to. It's all out there. So if you want to take the atlas and say extend it, you want to label new regions and rebuild the atlas, you could do it. If you wanted to, say, study the connection between the MR signal, which is more microscopic, and the microscopic images, the histology, you could also do it. You have this beautiful data set that is already aligned for you. The alignment is a nightmare, generally speaking. Also, if you want to, again, to use this for educational purposes, where you want to localize brain regions using the MRI and then look into them microscopically, you can use the beautiful viewer that you guys built, which is super cool. I love this tool. So that's the, so to speak, the data aspect of it, which is a big part of it. They're not cheap to acquire. It takes a lot of work and a lot of time.
Indeed.
Yeah. And also the reconstruction, the 3D reconstruction was incredibly, it took us a long time to figure out. So that's the data aspect of it. But then, of course, we have the atlas and the segmentation tool. The segmentation tool uses the atlas to automatically find all these regions in 3D images of, living people, or also like ex vivo scans. So for ex vivo, which is also kind of expensive because, again, putting a brain of a donor in the scanner for a week, I don't know if it sounds easy, but it's actually not that easy. First of all, it's expensive because MRI scanning is expensive. And it also requires certain expertise. If, so to speak, if you put a a dead brain in the scanner and press the button, it doesn't work because of differences in the brain. You have to put it in formalin, and then the tissue properties change, and normal MRI doesn't work. So it requires some expertise. So, but nevertheless, ex vivo MRI is becoming more popular, and also there's more and more data sets that are becoming publicly available, which is great. But the thing is, part of the reason why people didn't do that much before with ex vivo is that said, okay, I can put a brain in the scanner for a week and get these super high resolution images, but I don't have the software to take advantage of the high resolution images because all the software we have is catered towards in vivo scans, which are 1 millimeter resolution, whereas ex vivo is like 100 micron or 150 micron. With NextBrain, we can really squeeze all the information in these images because NextBrain is defined at 200 micron resolution and it has like a few hundred labels. So I'm hoping that really ex vivo MRI analysis takes off, or partly thanks to NextBrain. But the other thing is, even in brains of living people, even with limited resolution, you can still use NextBrain and identify all these regions that you couldn't find before. This is essentially the motivation for the original hippocampal atlas that started the whole thing. I like the hippocampus. It's a very good example because it's connected to so many things, right? Over a century ago, my fellow countryman, Santiago Ramón y Cajal, have you seen these Cajal prints?
Yeah, I probably wouldn't pronounce it like that, but probably totally wrong. But I know what you mean.
Cajal, yeah.
I know the purple or violet stem cells. No, they're fascinating.
But Cajal was a fantastic, he was really good at drawing himself. So he drew all these beautiful prints, right? And if you look at the prints of the hippocampus, like Cajal already knew that the hippocampus was not a blob, so to speak. The shape is not a blob. The shape is, as the name tells, hippocampus is a seahorse in Greek. So it looks a bit like a seahorse, right? But it's not homogeneous. Like Cajal already knew that the hippocampus had a shape. It rolls a bit like a Swiss roll. It has different subregions that have different connectivity. These subregions are affected differently by aging, by disease. We've known this for a long time from postmortem studies and animal models. But the idea is like, can we look at these things in living people using MRI? Previous packages, they can find the hippocampus, they can segment the hippocampus and label it, but just as a single blob. And now we can look at all the subfields. And for example, if you're trying to measure the effect of aging, or maybe the efficacy of some therapy, right? You give someone a drug and you want to see if it's slowing down Alzheimer's or something. You can find much more subtle effects by looking at the subregions than looking at the hippocampus as a whole. So now we can do this for the whole brain, essentially, right? Not only the hippocampus. We can do this with the amygdala, we can do it with the thalamus, we can do it with the hypothalamus, with the brainstem. So my hope is that people will start running analysis with this tool that is much more fine-grained and can help you find more subtle effects.
Have you actually got feedback from people who are using it, or have you known of people downloading and using it for their work, educational or research purposes?
Oh, man, it's too early. The paper came out like 10 days ago.
It's never too early.
I mean, there's already some questions in the mailing list, but they're mostly about, I don't know, someone who had a problem with PyTorch libraries or something.
Oh, right.
But I can't wait. Maybe you should invite me back a year from now and see how it goes. Actually, a colleague asked me to run it on folks who have undergone, oh my God, can I pronounce this properly? Hemispherectomy, like when they remove a whole hemisphere in their brain, which looks really crazy. When you look at the MRA, you're like, can this person be alive? So we actually started running it on these images, and it looks pretty promising. I don't know what we will, it takes a while. I don't know what we'll find, but you can analyze them, and it looks super cool. So there's definitely a lot of interest, but we need to wait a little bit to see what.
Okay, but it's hot off the press as we speak.
Yes.
And the other question that I have really is, now that I guess that the project itself is now finished, in the sense of it's being published and all of that. So how can you make sure that the software actually lives on, because that needs to be maintained and all of that? How do you keep the lights on now that the funding for that particular project has ended?
Right. NextBrain is now part of FreeSurfer, which is one of the big software suites in the neuroimaging world. There's like 4 or 5. There's FreeSurfer, there is FSL from Oxford, there is SPM from UCL, actually. There is one from the NIH called AFNI. So there is like a handful of packages that essentially every neuroimaging lab in the world has on their cluster or their computers.
Oh, right. Okay.
Yeah. So FreeSurfer has been living for a long time. It's going strong. And there is enough people contributing to it and depending on it and asking, like, writing grants to putting together tools that go into FreeSurfer, that I'm confident that it will be fine. Every time we write a grant to build this type of tools, we always put some dedicated money. We make essentially FreeSurfer integration and maintenance an aim or a sub-aim of the grant.
Oh, right. Okay.
So we put money for software engineers in the budget. There's enough people doing this that the software, I'm pretty confident, will live on. I'm doing this myself. Like every time I write a grant proposal for the NIH, aim 3.3 or 3.2, whatever, is FreeSurfer dissemination and maintenance. And we put some dollars in the budget.
So now that the project is finished after 10 years, what are you going to do next?
Oh man, I don't know. I feel a bit like this Comedy Central or Adult Swing or something where the coyote finally gets to the roadrunner and then he falls in a depression or something because he doesn't know what to do anymore.
You don't strike me as somebody who falls into a depression.
Felt a bit like it. Funnily enough, what happened was when we were working on NextBrain, we thought we're going to have this atlas at ultra high resolution, and we're going to use it to analyze ultra high resolution MRIs from dead people, but also, like I said, MRIs of living people. The MRIs of living people are going to have a resolution that is lower than the atlas. That means that there is a resolution gap that you need to bridge. So we worked on these machine learning methods that would be able to bridge this gap. And this is science. You never know how things are going to work out. Turns out that they were okay to bridge that gap. But if you shift the resolution to the right, so to speak, so instead of saying like, okay, the gap from ultra high resolution ex vivo, yada, 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 to in vivo, you actually shift and say, no, let's go from in vivo research grade. So in research, we acquire these beautiful isotropic images where like essentially so a voxel is a pixel in 3D, right? So the voxels in the scans that we acquire for our research studies are isotropic, meaning they're like cubes. You can think of a little sugar cube. But in the clinic, they don't like that. What they prefer is saying like, no, instead of having like, imagine like a Rubik cube, but instead of 3 by 3 by 3, it's like 200 by 200 by 200. If you wanted to inspect the whole thing, it would take forever because you have like 200 slices to look at.
Yes.
So in the clinic, what they do is that they acquire 25 slices with very high resolution within the slice, but a lot of space in between them.
And then they try to bridge the gap between the slices.
Exactly. So the thing is, computers don't like this. Computers prefer the Rubik cube, right? They have no reason to prefer one orientation over another. So for 3D analysis, these like isotropic cubes are better. So until recently, we didn't have tools that could analyze the clinical data effectively. So it turns out that the tools that we originally worked on to bridge the gap between ultra high resolution ex vivo and these isotropic research grade scans turned to be super effective, like really good, shockingly good. I never expected this, at bridging the gap between the isotropic research grade and the clinical data.
But this is where the surprise in science comes in.
I know, yeah.
You know what happens when you do that.
They are so good. And the same with portable MRI. So this is a technology that is more or less recent. You have a similar problem where the voxels are fat. They're not like thin and small and isotropic. So it turns out that all these methods that we developed are really good at these related problems. So a lot of our research now is going into trying to come up with machine learning tools that can analyze MRI scans of any resolution and contrast. So it doesn't matter whether the slices are parallel to the ground, parallel to the wall, or parallel to the other wall. No matter if the spacing between them is 5, 7, 6, or 2 millimeters. No matter if they use different types of MRI contrast. There's different modalities of MRI. I don't know if you've heard, like there's T1 weighted, T2 weighted, flare, proton density weighted. So we are designing tools that can swallow any of these scans. And the cool thing about it is that if you want to study, say, Alzheimer's on white males like me, there's plenty of data sets. There's this database called ADNI. You have the UK Biobank. There is plenty of data. But if you want to study, say, a rare disease, or maybe you want to build a model for some minority, ethno-racial minority or something. Now what you can do is you can go to the picture archival system of your hospital. A minority is a minority, right? But if the N is big enough, and like Mass General has like a gazillion scans in the database over the years, you can pull those scans without having to scan anybody.
And analyze them.
So we are super excited about that. And the same technology actually works very well on portable MRI, which we could do a whole podcast about. It has like massive upside in underserved areas, like say the middle of Ohio, where maybe not Ohio, but like middle of Montana, where the closest MRI is like 4 hours away, or even say Mass General Hospital. If you have someone in the ICU with a stroke and you want to scan them, but moving them to the basement is actually compromising their health or their life,
Yeah.
you can actually bring the scanner to the person and do point of care imaging. But the images are, for the lack of a better word, a bit crappy. So these type of tools are really cool. So this is what my group is focused on now.
Okay. Well, thank you so much, Eugenio, for your time today. That was really exciting. And maybe there are some follow-up podcasts that we can do in future.
Thank you so much. And very quickly, I would like to thank my whole team, both at UCL, here in Boston, and all my collaborators that made this happen, because it was really, really, really a team effort. Like I was telling my colleagues the other day, some of the middle authors in this paper have done more work than first authors in other papers. Like, it's been insane. So I'm really glad it got published in a really good journal. I felt that, I owed it to, that includes you, by the way.
Yeah, I saw my name on it. Thank you very much.
No, but seriously, I'm so happy and relieved.
As I mentioned at the beginning, James Hughes and I worked together on this project, and our job was to develop a browser app to navigate through the brain in all its different projections and resolutions. James did by far most of the work on this and did a brilliant job, quite frankly. And I wanted to talk to him briefly about his work, how he approached it, and what he took away from working on it. So here's James.
I'm James Hughes. I'm a senior research software engineer, currently working at the Advanced Research Computing Centre at University College London. I particularly specialise in building web applications for researchers. So that includes everything from designing and building research user experiences, healthcare-related applications, in-house hospital apps, but also more bespoke scientific applications, which I guess relate more to what we'll be discussing today.
Exactly, that's NextBrain, because that was a web-based application. And I remember that we worked together on this. I mean, you did all the work, to be honest. But, so I was supposed to be managing the project. But anyway, so could you quickly describe what your aspect of the work actually was? I mean, NextBrain has several components, but yours was actually web-based, wasn't it?
Yeah, so I was responsible for the implementation of the application itself. So that was the actual web-based components, the tech stack, the architecture, and basically translating what the researcher's vision was into a usable web application, in essence.
What I remember is that we had MRI images and then we had histology images. The job was to go from one to the other and switch between the two, wasn't it?
Yeah, exactly. So at its heart, the NextBrain web app is essentially an interactive brain atlas that allows users to explore the brains of 5 human donors. I guess what makes this particularly interesting is that the app allows users to navigate synchronized views of MRI scans alongside very high-resolution images of histology slices.
And that brings us to one of the challenges that we had was that the slices were actually enormous, weren't they? I mean, the original data set was huge because, they were very high resolution images.
Yeah, for sure. There was definitely a challenge around dealing with the complex data structures. So we had a constraint in this particular project in that the anatomical brain region data was stored in compressed multidimensional NumPy arrays. And I'd say that isn't really a typical data type that we usually work with in front-end web applications. And then on top of that, JavaScript also doesn't have built-in support for complex matrix maths either, certainly not like languages such as MATLAB, for instance. So yeah, there was definitely a significant challenge in developing creative solutions for working with these particular data types in a web environment.
So how did you get around that then?
We actually ended up contributing to some open source software for this particular project.
Which one?
A package called NPY.js. And this essentially allowed us to more easily interact with these multidimensional arrays. under the hood, do all of the calculations that we needed to do to calculate exactly where in the brain we currently were and how that translated to the location of the histology slices as well.
But these were not the only challenges that we had, weren't they? So there was the mapping, the translation of, well, not a translation, but the dealing with NumPy arrays, multidimensional NumPy arrays. I think there were a couple of other challenges, weren't there?
Yeah. So as always then, one of the main challenges in any complex project really falls around communication and accurately translating the researcher's vision into a user-friendly application. And I'd say this was especially important for this project, given that we had a rather limited budget to work with in a timescale of around 3 months of work. So not long at all, really. And coming into this, I knew there were going to be unfamiliar neurological concepts, domain-specific knowledge, as well as trying to understand the complex brain segmentation data. So I remember one of the first things that we did on the project was to have a series of in-depth conversations, just chats with the researchers to establish a level of communication that could really break down any barriers or blockers as quickly as possible. But leading on from those initial conversations, as I said, the next challenge really was trying to actually figure out how to implement the data that we have within a web environment using the most appropriate software architecture. So I knew we needed an architecture that would allow the data to be visualized seamlessly and quickly. So in the end, I remember we decided to build this as a pure client-side or front-end web application. And I think that was a really good choice on reflection because it really did make the synchronization between those brain segmentations much, much easier to work with. The tech stack itself, it's a typical React TypeScript application, which does typically work well in producing these kinds of slick user experiences.
And that was quite quick, given the time frame, we've actually got something going pretty quickly. And also what surprised me was the fact that the loading of these huge histology images actually worked quite well. But I think we used a trick there, didn't we?
There's a couple of things that are going on under the hood to allow that all to happen really quickly. The first are the data structures themselves. As we said before, we have these multidimensional arrays. But then there's also the user experience itself. So by default, all of the images that the user sees are rather small, compressed images, but still, detailed enough to see what you need to see. But then the user also has the option to select much higher resolution versions of those images so that they can really zoom in on the details and see all of the smaller cellular structures in the histology slices.
Looking back, what are the lessons that you've learned from this project?
I think a major takeaway from this project for me is that it really taught me to embrace the unknown and just kind of face it head on. As we've said before, at the start of the project, it did present some intimidating challenges, especially since at the time I was still a fairly new RSE. But in the end, when you kind of look back at what's been achieved, you realise you got through all of those challenges. And it makes you proud to have pushed through and not given up. And finally, I think what made this project really effective is that we all managed to kind of foster this non-judgmental and supportive environment together with the researchers. And that's actually something that I've taken and tried to apply to all of my projects ever since then.
As per usual, you'll find the links to the project and the paper in the show notes. Next week, we're going to the city of Newcastle in the northeast of England, UK. Newcastle has been hosting the Research Software Engineering Conference in the UK not once, but twice, in 22 and 24. Apart from Newcastle being an exciting place to be in, it is also the home of a growing team of research software engineers. And I had the great pleasure of meeting some of them once again at the end of 2025. And in the next episode, you can find out what they're up to. I hope you'll tune in then. And in the meantime, thanks for listening. Oh, time's up. See you next time. But before I forget, this podcast is covered by the Creative Commons license. See ya.